Welcome, everyone. I see that we have a good collection of people who are joining, and I am absolutely delighted to have our panelists today for a topic which I think is quite close to all of our hearts, in that things have changed a lot in the last year. And 6 months ago, we were talking about changes in the way we do clinical trials, doing them remotely. But now I think looking at some of the changing regulatory landscapes, it has brought very much into focus things that were probably already happening, but have been brought into focus much more so by the COVID-19 landscape. So we've got really tremendous cast for you of people who've got a lot of background in understanding the regulatory aspects of a whole range of different therapeutic areas, and also from both sides of the fence, from pharma and from agencies, particularly the FDA. So I'm really looking forward to this discussion, but I'm going to start with some housekeeping rules for everybody whilst people start to join. The first thing is that this session is being recorded. There are a number of people who have got in touch with us and said they can't actually attend, and we are recording it, so it will be available after we have completed, and will also be available through the transcript website. The panelists will all want to state, I am sure, that the opinions that are said here are the opinions of themselves and not necessarily the organisations that they represent, so that please don't hold that against whatever we might say, and we will be careful not to be too controversial. All the attendees are muted, and only the panelists are able to talk, but there is a chat box open, and I will be watching the chat box together with the other panelists. So if you want to ask questions, please put the questions in the chat box and we will endeavour to answer those. So I think that that's probably all the things that I've got on my list that I must say before we start. And I am going to ask the panelists to introduce themselves. Before they do, I am Flick Gabay. I am the managing partner of Transcript. I'm a pharmaceutical physician with a range of different therapeutic areas that I have been involved in big and small pharma over the last nearly 40 years. And I am also much involved with the Faculty of Pharmaceutical Medicine, which has been looking at a lot of these issues recently. So I'm really intrigued to hear what our panelists have to say. I think it's a great area for discussion, and we're going to start off by them introducing themselves and talking a little bit about how they think things might change from their perspective and what the key issues are. So we will start with Ed. If you could give us a little bit of background, and we'll go from there.
Great, thanks, Flick, and thanks for the opportunity to join you here today. I'm Ed Cox. I'm in regulatory affairs at Regeneron Pharmaceuticals in Tarrytown, New York, in the US. I'm an infectious disease doctor by training, and prior to joining Regeneron, I worked at FDA for over 20 years, mostly in the area of infectious diseases. And as Flick mentioned, the thoughts I'll share today are my own and don't necessarily represent that of my company. And really my intent is to keep the discussion academic and not product or company specific. And Flick, you wanted us to make a couple of comments about sort of, where we see things now and where we see things going and how things might change in the future. So as I mentioned, I'm an ID doc, an infectious disease doc, and when I think about emerging infectious diseases, I actually back it up to 1918 flu, and then think about the original SARS, and then HIV, 2009 H1N1 influenza, the outbreaks of Ebola that we've seen over the last, several decades, with some particularly large outbreaks over the last couple of decades. And SARS-CoV-2, COVID-19 is obviously, a pandemic beyond, what we've seen in emerging infectious diseases recently. You got to dial it all the way back to 1918 to get to something of similar global impact. And I expect that what we'll have here from our learnings from COVID-19 will be incremental learning and incremental change. I think there are some tools that we're seeing that are probably more prominent and more recognized now that we're in COVID-19, such as the use of master protocols, adaptive designs. But I think, as we'll hear from some of our other panelists, they're not necessarily new. We actually have used master protocols probably first in the world of oncology, as we'll hear from some of our colleagues, and also in infectious diseases. The trials that looked at therapeutics for treatment of Ebola were designed as master protocols with the ability to study multiple therapies. So some newly recognized tools that are taking on greater prominence because of their utility in the setting in which we're studying therapies for COVID-19. We're also grateful that there have been technological advances that help us in developing therapies and vaccines, and also technological advances that impact our ability to perform clinical trials, such as the ability to monitor patients remotely, and some of the new tools that, in essence, are present not only in the world of clinical trials, but also in our daily lives as technology advances. And so, just to sort of sum it up, and where does this all end up in the bottom line? Well, for me, I think what's really important is to select the scientifically appropriate tool or study design or endpoint for the disease that you're trying to study. And that's going to depend upon the nature of the disease, the nature of the therapeutic, and the conditions under which the therapy is being studied. So always important to pick the scientifically appropriate tool. And when you look in the toolbox, there may be some very shiny tools, and it may be tempting to grab that shiny tool. And the shiny tool may sometimes be the right tool to use, but other times the tried and true one that's a little worn may actually be the best tool for studying your particular disease or therapeutic. So it really is, I think, about picking the right tool for what it is that you're trying to study. So I'll stop there, but thanks for the opportunity to make some initial comments, Flick.
Thank you. And then I think we're going to Renee, and maybe you're going to talk a little bit about something completely different for a lot of us in terms of Galvani and Alphabet. So over to you, Renee.
Thanks, Flick. Good afternoon, everybody. I'm Renee van der Merwe, and I'm the Chief Medical Officer at Galvani Bioelectronics. So some of you probably don't know much about us, but we're a device company looking into bioelectronic medicines to treat chronic diseases, especially immunomodulation. And it's formed from a partnership with GSK and Verily Life Sciences, which is part of the Alphabet Group. And I think what's really exciting to me, I spent the majority of my life in pharma, the majority of it being within MedImmune, AstraZeneca's biologics arm, and subsequently, more recently, in a small biotech in Paris. But I think within devices, things are so different. You don't go through phase 1, 2, 3. It's kind of, you have a feasibility trial and you have a pivotal trial, and you don't have to do replicate pivotal trials either, which really opens up a lot of doors to use these new, well, not so new designs for us. And especially you're talking about platform trials, Ed, and I think definitely within our device and looking, within immunomodulation, platform trials really lends itself within devices. And because we have to show such huge efficacy effects, that it makes the design so much smaller and simpler. But the one thing to me, Flick, when you asked me about this, and we were having some discussions earlier, and based on what you've said, Ed, as well, to me, all these shiny tools and designs that aren't so new. Actually, it's just kind of where you are in the life cycle of development. And if you've been here long enough, you've seen things come and go and what's become fashionable. And it's literally like fashion. You just need a big warehouse to deposit all your data in and then bring it out when it becomes fashionable again. But I think, you know, to me, what has always stood me in good stead with a development plan is it's based on the science, the cost, and the time. It's that triangle. And depending on what you have in science, how much time you have, and how much money you have, you're going to have to flex on those different levers. And any development plan is absolutely right, Ed. You have to know what your question is. And depending on how much money you have and time, you can use really fancy tools or different tools. And I think it's just deciding exactly what Ed said, using the right tools to answer the question you have with those resources you have in mind. So for us at Galvani, it's really interesting. And to me, it's been quite a new experience, you know, just looking at what do I need to achieve this. It's not all these hundreds of patients that you need to expose. It's a very different therapy area. Well, not a therapy area, but a development area. And it's truly exciting. I mean, working with Verily and having not just the electronic expertise, but also the digital tools and recruitment tools. Absolutely, you know, they have an entire toolbox of tools as well. So it really is a fascinating journey. So I very much look forward to the discussion.
Thank you. That's really helpful. Kevin, you come to this from a slightly different approach from the statistical background of things, but actually which underpins so much of our decision making.
Sure. So that was really interesting to hear comments, you know, from Ed and Renee. So a bit of background and then just a few comments in terms of perspective. So I'm Kevin Carroll, a statistician in the industry for 32 years now. About 21 of those spent in AstraZeneca, where the last role I had there was the VP for statistics and chief statistician for R&D. So I had some oversight over all the R&D areas. And then I've been working as a consultant with small companies since about 2014. In terms of, you know, the topic of our WebEx today, I mean, at the end of the day, you know, I like to step back and ask myself, what is it that we're actually trying to do here when we're developing medicines or devices or biologics? And what we're actually doing, and we're not even selling medicines, we're not even selling devices. What we're doing is we're generating information, selling the information. And the whole process is about the accumulation of data and evidence that collectively enables an assessment to be made of the therapeutic value and obviously the safety of whatever the intervention is we want to bring forward, whether it's a device or a biologic or a tablet or whatever it may be. Drug development is this endeavor of accumulating evidence. But unfortunately, we don't think of it like that. You know, we have this arbitrary divisions of phase 1, phase 2, phase 3, which I don't think is really the way we should look at things. We should be thinking about the accumulation of evidence. At what point do we have, can we make the case that we have a worthwhile intervention available to us? And there's, from my point of view, there's far too much reliance on the p-value in early development. It's an absolute tyranny, the p less than 0.05. We're a hostage to a kind of mindset that says we need it. We're a hostage to journals who say we need it, when in fact we don't. You know, early development is about what evidence do I have? What is the accumulation of that evidence that suggests the drug or intervention may be effective or may not be? It's a weight of evidence issue. P less than 0.05, P greater than 0.05 in early development is meaningless in my view. And it's far too much. So I hope in the future that we can get a bit more mature about what is it we're trying to do. It's not about, you know, the dichotomy of black and white. And yeah, I hope that we can find a way of using alternative data sources, especially in things like rare diseases where there's just not enough information out there. Infectious diseases are terribly burdened by the notion of, you know, if you want to do a non-inferiority study, well, how do you, well, your margin has to be super small, your trial has to be super large, because nobody's done any, you know, placebo-controlled trials, infection or pneumonia for 70 years. So when we try and apply the regular way of doing things, or the more common way of doing things, I think we do that inappropriately in early development. But there does come a time, of course, when you've accumulated your evidence, that there has to be, the evidence has to still pass the critical test of, is it reliable? Is it truthful? Is it unbiased? Can I look at this evidence and draw a fair and correct conclusion? So for that reason, you're never going to get rid of randomized trials. They're going to have to be part of that evidence base, because they're the only real things that can really tell you for sure about the true underlying efficacy. So it's going to be part of your evidence base, but nevertheless, the onus is on the sponsor to create that. And I think the old ideas of phase 1, phase 2, phase 3 should be gone. It should be, what data am I generating? What more do I need? And we should, we should throw off the shackles of that p-value. It is a disastrous notion that in some ways has been introduced by statisticians for such a long time. I mean, and I'll close by just making a small comment about p-values. I'm not sure how many people know this, but the question is why? Where did the p-value come from? Why is 0.5 supposedly this magic dichotomization between good, bad, success, failure? Well, there is a story that goes along the lines of many moons ago, there was a lady who suggested that she could tell with a cup of tea whether the milk was put in first followed by the tea, or whether the tea was put in first followed by the milk, and she could tell which way. And in order to devise a test as to whether this lady was telling the truth, a test was devised, and the 0.5 level was used as the strength of evidence against that claim. And that, as far as I'm aware, there may be other earlier times that the 0.5 level came up, but the application of 0.5 in the context of figuring out whether your milk went in first or foremost, vis-a-vis whether some therapeutic should be made available to some poor old soul who is, desperately sick with breast cancer, for example, is ridiculous in my mind. So the 0.5 thing is just got out of control. It should be about evidence, and what evidence do we have? What's the weight of that evidence? And be sure it's reliable, unbiased, and meaningful.
I think that's very, very real and strikes to the heart of everybody who has to look at the evaluation of benefit risk is being able to understand that. So I think that's a really good thing. Even if the tea analogy is particularly British or even English, actually, but a very good one. Giorgio.
Hi, good morning, good afternoon, good morning, everybody, wherever you are in the world. I'm Giorgio Massimini. I am an MD by training, and I spent the past 35 years in the pharmaceutical, working mostly in oncology. Past 3 years, I did some foray in immunology and in COVID, because everybody is working on COVID. And to your question, Flick, what has changed? Science has changed a lot. We have got a much better understanding of the biology of the disease, and we got also much more powerful tool to understand that. And this has a major impact, because our flexibility that we are seeing now in the regulatory world and in academic world is coming from the fact that this new knowledge brought a new question and a new way to address this question. If you think of the COVID-19 virus has been mapped in 6 weeks. That was a speed that was unheard of, and the same thing has happened in a lot of diseases. I mean, when I was a student, chronic myelogenous leukemia was a deadly disease that would kill you in 3 years. Now, a patient with chronic myelogenous leukemia has the same life expectancy than anybody else, thanks to the understanding of the biology and to develop the new drugs. And again, I think we had the discussion before, I saw the flexibility of the health authorities changing with the amount of drugs that were coming. The more drugs are there, the more options there are for developing, and the more options are for the health authorities to adapt their knowledge, because they base themselves on science and nothing else at the end of the day to make their decision. So they follow science. I fully agree with what was said before about the importance of the clinical program rather than the clinical trial, because it's a continuum. It's not something that you can't have a study as a standalone. It simply doesn't work that way. So the entire program has to be worked out and planned in a way in which the endpoints are meaningful, the studies are meaningful, and we are answering the relevant questions. Seems to be, I would say, a banality, but it's not. In COVID, I saw a lot of studies right now in which they went for the duration of symptoms and having a secondary endpoint, hospitalization and severe COVID. And the final result of the studies, probably Ed can comment on that as well, is that the study fails the primary endpoint and is positive on the secondary endpoint. So there is an issue there on how we look at the real critical question when we go for development. And I think that's the something that will change and probably will improve in the coming year. The other thing that is changing is the tool, but not so much the tool in terms of clinical trials, because we are using the same tools that we are using 20 years ago. Nothing really has changed. The way in which trials are designed didn't change much. Adaptive design are already available many years ago. What is changed is our ability of handling data. And what is changed now is our ability to collect data from the population. I mean, to me, what is going to change a lot in the coming years is the real world evidence. Because now we have access to an enormous amount of data, which before we couldn't use, essentially, because we didn't have the tool to do that. And if you think of, at the moment, they are complementary, as far as I can see, to the phase 3 trials. Because at the end of the day, the phase 3 trial, you have to remember at the beginning, was a confirmatory trials. So what better to confirm a confirmation that have data coming directly from real life with all the problems you can have in real life to confirm what you have there. Very recently, the FDA has evaluated 6 different indications in 6 different drugs, 4 different drugs, for 6 indications which they got accelerated approval. 4 were maintained and 2 were withdrawn at the end. The interesting thing is that all those drugs have been used for at least 2 years before they came to this point. And they came to in front of the ODAC because the phase 3 that was designed as confirmation trial didn't meet the endpoint. Very interestingly, none of the pharma presented data coming from real evidence, even if there have been hundreds of patients treated with those drugs in late stage of the disease, of course, before they came to the other. So even the industry has to make a step to understand how to handle these things. And probably Renee, working with Alphabet, has much more information about this that I have.
Thanks, Jojo. I think that's a good place to stop and to go into more of a discussion. And I think I would like to bring you back, Ed, in the sense that even when you were still at the agency, 60% of the drugs that were being registered were being registered for orphan or rare diseases. And in many of those, they would be as accelerated approvals or as unusual trial designs that were adaptive. And I think everybody around the group is saying that the science and the placebo control, the whole basis of evidence is really important in placebo controlled trials somewhere. But we are already accepting that as we go to the trials beyond proof of concept, that we might go into different designs. How do you think, and I'm not asking you to tell me how the agency will see it, but how do you think we might progress in future in the difficult part of phase 3, in inverted commas, in our good old-fashioned, well-controlled trials? Are we going to move much more to either real-world evidence or to adaptive designs? How do you think that's going to progress?
Right. So yeah, obviously I can't speak for the agency. I don't work for them anymore, but just some comments and some thoughts on the issue in general. Again, I really think it is, picking the right approach depending upon, what you're proposing to do. And, if you're considering, looking at 3 doses and carrying them forward into phase 3, but, you want to do, a look at some point and decide, do you want to continue on with 2 or 1 of the doses based on some criteria that you've set ahead of time, an adaptation in the trial of that nature, that may be appropriate in some circumstances. And really, what matters a lot too is the nature of the disease. In some diseases, the progression of the disease is relentless and there's not much variability. I mean, you can pretty much predict what the outcome will be when you look at a population of patients. In other diseases, the outcomes can vary tremendously. You can have some patients, that have mild disease, some patients that have very severe disease, some patients who progress on to fatal disease. And sometimes you can measure some of the factors that impact the likelihood of those different outcomes, but you can't measure them all. And that's when you get into a disease condition like that, and many acute infectious diseases are, can be of that nature, it can be challenging to, to predict what the outcomes will be for a particular group of patients, which is why, you really do need a control group in order to make, good, good appraisals of how your drug is performing. So, a lot depends, I think, on the nature of the disease. And so, we've heard mention of real world evidence, and the term means a lot of different things to a lot of different people, depending upon the context that it's being used. But certainly using real world evidence, trying to understand the natural history or course of disease can be very, very helpful. Trying to understand, how a therapy may impact a disease in sort of a more sort of gross observational sense may also be possible. And if you have a disease where you can actually reliably use an external control, then real world evidence, can probably be quite informative. If it's a disease where you really do need to have a concurrent control generated by randomization in order to make sense of the data, then real world evidence, it's going to be more challenging. And in that setting, you may want to incorporate randomization into real world evidence. Typically, that would be in the setting of a drug that's already approved rather than an investigational drug, though, too. So that's some thoughts on the issue. And I want to mention one more thing too, which is a theme that I think I heard, throughout all the comments, and that is the issue of benefit risk and how do you balance benefit risk depending upon the nature of the circumstance that you're dealing with. Are you dealing with a serious disease where there's no available therapeutics? Are you dealing with a less serious disease? They're generally mild and patients experience symptoms, but they all get better over time. And there's multiple different therapies available. So you can see I've painted a spectrum of disease severity, a spectrum of degree of unmet need, and how you approach those situations is impacted upon by the nature of the seriousness of the disease and the degree of unmet need. So just one other theme that I've heard in my co-commenters' comments.
Thanks. Renee, I'm going to come back to you about the real world evidence, inevitably, because for you and being linked to Alphabet and so on, this is something that you may be able to use. And I think certainly when you've got things that are a kind of a device where you've got surrogates that might be thought to be scientifically proven or not, as the case may be, and then you're looking for real world data to support what you have. How do you approach that sort of thing?
Sorry, Flick. I think for us, it's definitely, going back to science, because science is going to drive everything we do. And then, I think the next step is, once we know what questions we need to ask with the science and what we're short on, especially in our device world, with implantable devices. I mean, there are neuromodulation devices out there for migraine and sleep apnea and likes like that, but not for immunomodulation, and anti-autoimmune diseases. And I think the key for us is just looking at what would immunomodulation do on top of anti-TNFs or JAKs or IL-23, 17s, any biologic with that. And then looking to see, Ed was just saying, and Giorgio as well, real world evidence really does come into play with a number of, depending on what disease you're looking at, there's so much data out there that it's kind of silly not to use it. Because once you add it into a clinical trial, first of all, it's the cost of giving that biologic on top of what you're doing, which adds an astronomical amount to cost. And although we own by, a partnership of GSK and Verily, it's very much run like a biotech company. We have a limited resource, so we have to make do with what we have. And one of the key things we've been looking at is using the Verily data set for real world evidence and having single arm studies. And of course, it can't work for every indication we want to go ahead, but for the majority of indications we want to go ahead in, it definitely does help. And, working out a development plan, cost and time, it adds considerable benefit.
Yes, I think so. And I think also one of the things that Ed was reflecting on there is using the data to understand the background where you're going. And I think that using real world data to understand disease process, so that you can understand a lot of the things that Ed was referring to, like what the outcome might be and what you might expect, because that hugely influences not only your trial designs, but your program designs. And particularly, I think, Ed, the variability of patient outcome, which makes a big difference to some of our trials. And I think, Kevin, one of the things that we do find quite difficult, I think, with the statistical things, is where we've got diseases with big variability of outcome or uncertain outcomes, then we have real challenges. And that's often where the phase 2 / phase 3 challenges come, is choosing the patient population for phase 3 if your phase 2 outcome is not as clear and there's a big patient variability in the phase 3 studies. And I guess things like propensity scores and stuff tend to be used. But do you want to make a comment on that?
Yeah, I come on that. I mean, it's true. I mean, we're talking about real world evidence, I think, and it's true that there's a lot of data sources externally. The danger, though, is that might be an awful lot of data, but an awful lot of imprecise or incorrect data, because the fundamental issue at hand remains whatever evidence you have about your drug or device, whatever that evidence is, whatever it's coming from, trials, RCTs, single arm studies, registries, real world evidence, wherever it's coming from, the challenge is you have to be able to conclude from the evidence. You have to be able to make reliable, correct, truthful conclusions that are not hampered by confounding. So if you're going to attribute something to a medicine or a device, and you're going to say, well, this medicine improves survival by this amount, this medicine has a certain cure rate in relation to antibiotics. If you're going to make a statement of claim, you have to have the evidence to support it. And the problem with the difficulty with the simplistic use of real world data, I think, and there was a friend of mine who once said, It's not real world evidence, it's real weak evidence. I remember him saying that in a disparaging fashion. Because the problem is you can't attribute quantities, you can't attribute the effect of a drug on an uncontrolled set of data. You're observing things that may or may not be attributable to drug. And therefore, you may not be able to make reliable conclusions, you may have bias, you may not have the truth. So in the end, however you get, whatever data you use, you must be able to opine on reliability, bias, and truth. Otherwise, nobody's going to approve a drug. So you have to be able to do that. Now, real world evidence can be, or evidence like that can be helpful in my view in situations where you've got rare diseases, or in particular rare diseases, or where you have particularly well codified existing, say, registries for patients with outcomes. And then you can use methodologies like Bayesian augmentation methodologies, where, for example, you would have a randomized trial, but with only a small sample size of control and a much bigger sample size of active, but it's still randomized. So the concept of patient selection is dealt with because it's randomized. But then you augment a small placebo group with your real world evidence, your historical data. There's methods of ways of making that placebo group bigger, and special techniques called discounting and other things that will allow you to make comparisons then between your imbalanced randomization with the extra data thrown in. And that can be an efficient way of arriving at reliable conclusions about efficacy. The fundamental problem isn't the use of data or whether you should or shouldn't use real world data. That's not the issue. The issue is what conclusions can you draw, and can you draw conclusions reliably without bias, and can you make truthful conclusions about attribution of benefit? If you can't do that, then you're in trouble. If you did a badly conducted randomized trial, you can't do it, you're in trouble. If you're based on real world evidence only, you can't do it, you're in trouble. So it doesn't matter what the data are. What matters is what can you reliably conclude from it. And I think that element, no matter what we do, will change. The way we generate evidence, the way we look at it, the way we analyze it, can definitely be improved. But in the end, it always has to end up in the same place. Can I make reliable conclusions? So I can't, I get nothing. Yeah. So I'm open to the use of all sorts of data in all sorts of different ways, but it must lead to the same ending point, which is there's no point using tons and tons of real world evidence saying kind of looks like it's okay, kind of looks like it's a treatment effect. And somebody says, well, actually, no, there's 25 confounding factors you didn't allow for, so you go nowhere. So if you're going to do these things, you've got to be done sensibly with the goal in mind. Otherwise, I don't think you're really solving the problem. The problem is, what is the true benefit of drug? What's the true risk of drug? Does one outweigh the other? I think that makes some sense.
Yeah, no, I think that's, and I'm going to come back to you, Giorgio, because I think you work in oncology in an area where there's a lot of variability, and where actually there's a lot of change as well over time. Yeah. So the standard of care is sometimes dramatically changing alongside the variability of the patient and pool that you're operating in. And we've started using things like basket designs, and I can see Ed, you smiling a little bit, because we talked a lot about potentially using those in infection where we've got similar sets of variability. But Giorgio, how do you think with the basket designs, how do you then transfer into phase 3 in inverted commas, or into because you can't just go straight into real world data?
No, but
progress that.
We are going more or less in this direction in a way, because the critical point on this basket design is always the selection of the population. And that's addressed partly the objection that Kenny raised about the real world evidence. Because what we had in oncology originally was a very large variability, because we were coming in with chemotherapy. You treat everything, you destroy the DNA, you cross your finger, you hope it works. It did, to a point. What's happening now in the past, I would say 10 years, is that even more, is that we are developing more and more therapies that are targeted to the tumor. And at this point, the variability in the population changes because you are able to identify the tumor that will respond better to that given treatment. Already that reduces the variability of your efficacy and also safety in the given population. The drawback is that essentially we are moving toward a serial of rare or quasi-rare diseases, because the classical example is lung cancer. Lung cancer in the early 90s was an untreatable disease in which the only thing that did do something with was platinum, and the survival was 6 months. Right now, the survival is completely changed. You have a 5-year survival of over 30%. And the lung cancer is not one disease. You have at least 6 or 7 subtypes or even more of lung cancer that we see and treat every day. And it's increasing because we are discovering new targets and we are addressing these new targets. Now, the other way of doing things, and the phase 3 trial, for God's sake, are not outdated at all. We're still using it very much, is what was expansion cohorts. If you think at the development of the immuno checkpoint inhibitor, the phase 1 were the monsters. There wasn't a single one that was less than 1,000 patients, phase 1. Why? Because the efficacy was incorporated in the phase 1 at this time, and not only safety. And after the classical dose escalation, there was an explosion of different cohort of patients with different diseases that were treated. The efficacy was evaluated, and that was the basis for the first registration. Then came the phase 3. Again, the situation, as Ed was pointing out, was linked to the fact that we had lethal diseases where there was no or little treatment. The classical was melanoma. The only thing we had at the time was dacarbazine, almost useless, and the BRAF inhibitor, which had activity, but only for a period of time. With the immune checkpoint inhibitor, things changes, and automatically the authorities accepted this approach. They had some reservation because after this experience, the FDA imposed a rule for which you cannot have for each cohort more than 40 patients. And originally they were much, much more, I can tell you. But still, this rule is not fixed, because you can, in certain situations when you have rare diseases, and we have some example ourselves, expand those cohorts to a number that allows to have a certain level of confidence, going back to the unbiased and truthful results that Katie was mentioning, based on confidence interval and duration of responses that confirm that the drug has activity. Without a randomization, because the randomization is practically impossible. When you have a drug that works in 1 or 2% of the patient with a given tumor, you can't run the randomized trial. I mean, theoretically you can, but de facto is impossible. The only thing you can do is to look at historical data and use some of the technique that Kevin was proposing to compensate for the lack of randomization, but you cannot do anything like that. But again, one of the key points to me in the development is exactly the choice of the population. It's the first couple of inclusion criteria and exclusion criteria define where you are and define the question you're asking. And this is the key point for being successful and reduce the variability.
Yeah, and I think that to some extent that was what René was suggesting, that there's a lot of data out there that can support that. Ed, do you think any of these kind of basket designs and adaptive designs will come in? I know we had lots of discussions, which are in the public domain, about whether we can use adaptive designs for antibiotic development as well as COVID-19 type things. Do you want to talk a little bit about those bigger platform designs and how applicable they might be in other therapeutic areas.
In oncology, they're already there. And we already had example of drugs approved based on basket design. I mean, the most was the one from Loxo that was based on 50, 60 patients for midline tumors. It were extremely rare, but that was a success. And there are examples of this. But again, those are rare diseases with where the classical approach is impossible because you can't randomize 15 patients. It doesn't make any sense, especially when there is no treatment for the control. Can it be moved to other therapeutic area? Yes, there is no reason not to do it, but the question is how rare is the disease and how you can validate the data. Now, there is a trick in the basket design, which consists in adding an arm in which you use standard of care, whatever it is. And that allows to have at least not a comparison, but a reference arm for which you know what your population is doing with using the standard of care. It has been used in oncology in different situations and allows at least to, I would say, bracket the activity of any other drugs you put in the basket or in the platform trial you want to use. At that point, using that approach, then you can, if you have exceptional result, it might result even in a filing and in an approval. Otherwise, it can result in a follow-up study, which has much higher probability of success than a shot on goal, if you want, without having this information. It's an evolution of the phase 2 design, if you want, but it might work. And it might work in diseases like COVID or other emergency diseases, because it's allowed to have a fast response to at least some questions.
So that brings Ed. Do you want to talk about these sorts of designs in infectious diseases, COVID-19, antibiotics generally?
Sure.
Where do you think we might be going?
Yeah. So, I mean, obviously, for COVID-19, master protocols have been utilized, and they have served an important role in studying some therapeutics. And it was interesting. Folks may not have caught it yet, but the FDA actually just issued a guidance document yesterday that came out, and it was apropos that Giorgio was making his comments about master protocols, because the COVID-19 guidance document draws on the previous oncology guidance document on master protocols and how they are utilized. And it really, the guidance document provides a nice listing of the different things you need to think about if you're considering a master protocol. There's the scientific issues, the design issues, the statistical issues, and then also the logistical issues that all sort of need to be weighed as one's considering a master protocol. So, it's hard to, I don't know that I can predict, how things will go, but I think the master protocols do provide an important option that's out there. And I think, critical to either, selecting, this approach is to really fully understand, all the different pros and cons. And the FDA guidance document does a really nice job of outlining these. And again, it gets back to picking the right tool for the question that you're trying to answer and then the disease that you're trying to work on. So
clearly something that actually could be extended beyond things and
yeah.
beyond oncology, and obviously we have seen some in use in COVID-19. What has got very confusing in COVID-19 is where we use those, because we also need to have some sort of robust trial, if we can, alongside some of the more adaptive big studies, in order for us to understand where the drug might be positioned. And as we've learned about COVID-19, there are many different phases of the disease, and some patients will not get symptoms at all. So different places of positioning does require some evidence in the first place, I think, in that respect. I don't know, Kevin, if you want to come back in on some of the more issues around
Sure.
adaptive designs and baskets.
Yeah, so just so we're clear, we use master protocols basket designs. I mean, those designs, and yeah, they're kind of born in oncology, but they're just screening designs. They're design where you are allowing yourself to expose different genotypes, different phenotypes to a given
Yes,
therapeutic to see what happens. So in oncology, it works reasonably well because there is a basic premise that tumors don't respond or don't shrink by themselves. You don't get spontaneous improvement in tumors.
Yes.
Now, whether that's true or not, we can debate that all day, but that's what that's built upon, and that allows the regulatory authorities. Incidentally, there's a huge difference between EMEA and FDA here, because the EMEA will not tolerate the notion of approvals or whatever it is on response in single-arm studies. But FDA do, because they have this kind of belief that tumors are not going to kind of spontaneously improve. And that allows you to try and see where you may or may not be seeing a certain response rate. I do agree, Giorgio made a good comment, which is these trials are much, much better if you do have, as one of the baskets, a standard of care arm. Because then, and I don't agree that you can't randomize. I just don't agree with that. I've done randomized trials with patients having, being drilled in the head and having stem cells delivered into the brain in Parkinson's, and we randomize that. So you can randomize studies, and of course we don't know if the drugs work. That's the reason we're doing trials. We can't presuppose we already know, otherwise why trials. But I think basket trials with some element of control are going to be far more useful in the context of trying to get a medicine to a patient, which is this is all about, because that gives you the opportunity to actually look at attribution of benefit. But adaptive designs, basket trials, they've been used for ages. They've been around since the 90s. They're certainly adaptive trials. And there's FDA guidance for both, and EMEA for both of those for adaptive designs. What unfortunately happened over during the noughties and the early part of the second decade is a poor application of those approaches. People think they can just do what they want. It's adaptive. We can look when we want to and see just subsets. And of course you can't, because you end up with biased results. So a correct application of adaptive designs can and do greatly facilitate development and can and do result in full approvals. I mean, some of the approvals I think that can happen in oncology and single arm studies are often just accelerated. They're only conditional. You have to come forward with confirmatory data. So that creates a whole other dialogue with the agency. But adaptive designs, if done properly, are usually controlled, and they can usually serve the purpose of generating a full approval and not just an accelerated approval if they're done properly. Fortunately, many in the past have not been done very well. It kind of left a bad taste in the mouth, I think, of FDA and EMEA. So if we're going to do them properly, and they're not free lunch. Really properly.
Kevin, totally in agreement. Two simple comments. First of all, when I'm talking about basket design with the standard of care, those cohorts are randomized always, because we are talking about a type 1 disease, essentially. So the patient are randomized across that, so the randomization is preserved. On your point about the tumor do not regress. In fact, they grow personally all of a sudden. It's not a continuous growth. But there are at least two tumors that have a very small percentage of patients that have spontaneous remission. Is a very old paper. Is from Lister 1980.
The exception that proves the
rule.
rule.
It tells you how old I am. But the two tumors are melanoma, where you have a 2% percentage of spontaneous regression. They exist. And the other one is renal, where you have 5 to 6% of patients that at the time experienced a spontaneous regression. But those are data that are going back in the 80s. After that, fortunately, we got drugs that worked, and at that point, the issue was put to
rest. I think that illustrates. I'm going to draw a little bit to a close here, because I want to talk about one other subject towards the end, which is that we've talked a lot about, we've agreed a great deal about everything. There needs to be robust evidence. And I think everybody agrees with that. Wherever we get it from, as much as possible, we need robust evidence to make that benefit risk. I think what we have seen in COVID, which has allowed us to use different tools more and so on, is collaboration. And it isn't just a collaboration between different farmers, but between farmers, academics, regulatory agencies, and so on. And I would like just a very brief word from each of you, probably starting with Renee. In the future, it looks to me like agencies are now saying to us for COVID, come as soon as possible and we'll have a dialogue about the program. How do you see that with the sort of program that you're running with a device?
Well, I must say it was quite an eye-opener to me when I joined Galvani. The interaction we've had with the FDA has truly been teamwork, backwards and forwards, ad hoc meetings, questions going backwards. It's something I've never actually experienced in pharma. And, their comment has always been throughout our whole process, come as early as possible. And it truly has been a collaboration. You were saying collaboration between academics as well. Definitely where we coming from, being absolutely pioneering different technologies, we have to collaborate with academics, and we have to identify that group of patients or physicians who truly want to go above and beyond the next TNF or JAK and help their patients. So for us, it's really all been about collaboration.
Ed, what do you want to come in on that?
Sure. Yeah. Yeah. No, I agree with Renee's comments. I mean, it's really important that that the enterprise that's required to bring a new therapeutic or new intervention forward are working together in a collaborative fashion in order to advance its development. And the discussions that you have with academics, regulators, people in industry, patient groups, I mean, the full continuum of folks that are affected here are really important. And I think if you're able to listen to all the different viewpoints and take them into consideration, there'll be a lot of good ideas that are generated from that. A lot of things that you might not think about independently that are gained from having these discussions across different stakeholders that can have really beneficial effects for a development program and help to increase the chance that a development program provides the information, the type of information that it needs to, and addresses important questions that are out there and increases the chance of success.
No, I agree. Jojo, briefly.
Same comment. The issue of collaboration is being addressed, I would say. I had the same experience with the FDA that Renee had in the pharma, because depends from the decision you're working on. You have different approaches, but some division of the FDA are very collaborative. The WHO is fostering as much as they can collaboration for the treatment of COVID, and their solidarity drive is an example of that. So yes, it's at the end is the basic of science. Science is discussing hypothesis, data, and reach conclusion. Not everybody will reach the same conclusion, but at the end of the day, is what we have to do every day.
Yeah. Kevin, and then I would wind up.
I would have little to add to what Renee, Ed, and Giorgio had to say. I think one has to be a little cautionary about COVID, the situation with COVID, and the extent to which regulators, EMA, FDA, said, please come to our door quick. That's an unprecedented situation. Whether you would see the same depth of desire for serious rapid engagement and serious decision making quick in the future, I think is unlikely. COVID is strange, but nevertheless, early engagement with the regulatory authorities is essential. If you don't do that, and the broader groups that I had mentioned, if you don't do it, then you're going probably to find yourself hitting some rocky waters, but some rough waters. But I think one of the things I found, it depends on who you're dealing with, which agency is it, which division within the FDA. And sometimes I do worry a little bit about the sheer volume of development requests, consultation requests that go to the FDA, and can they actually support that? I mean, it's crazy the amount of work they have to do. And sometimes you get the sense, things are a little bit rushed because, I don't know whether they just have enough people on the ground. Ed will know that better than me, but it's a sense I get. Sometimes I think, gee, this is tough because we're asking too much. So, but in principle, of course, collaboration is absolutely essential. You need it. If you don't engage in it, you're going to find yourself in trouble later down the line.
Right, so I think we're out of time, unfortunately, but I think the answer that we have addressed here is that we are not locked into particular trial designs, particular tools, and that actually what we are locked into is a good scientific basis of making decisions, choosing the right endpoints, the right patients, the right trial design, and that it's not going to be the same for all of the studies or all of the programs, and that early collaboration is really important, and that we need to be aware of what's going on around us. And I think that's been one of the most tough things to do. I think all of us who are involved in COVID would say that that has been really hard, to stay on top of the volume of literature that is being produced and guidelines and so on. So I would like to thank our panelists. It's been a very good discussion, and thank you very much for taking the time, and for our people who've joined us to listen. And go away, think about new exciting program designs, and discuss them, as Ed says, with everybody you can. Thank you.
Thank you, Flick. Thank you, guys. Very nice discussion.